So welcome everybody to our synthetic biology webinar. I apologize for being late. As you know, we have something coming up in a short while called SynBio Beta, and we have the biggest speaker that we've ever had that we're going to be announcing on the stage. So I'm going to be announcing that probably tomorrow. So that was what I was busy dealing with. We wanted to talk about the SynBio Beta Biopharma report that was released just this morning. I have a draft copy in front of me that I was going over with Mark last night, and I'm super excited about this. So welcome Mark Bunger from Innovation Lab, and thank you for helping us put together this report. So we're going to be going over the Biopharma report, and we're going to be talking about biopharma and the industry of the biopharma industry, and the market segments, the size and the history of it. We're going to look at some of the non-corporate actors that are coming into the industry, and some of the key factors that show how the industry is growing and what are the key factors that are affecting that growth. And in particular, then we're going to start looking at the value chain structure and how the different pipeline goes to getting a drug to market, and why it's so difficult to make drugs. And in particular, that's relevant to the synthetic biology industry, because the synthetic biology industry is important because it's trying to make biology easier to engineer. And so the goal of trying to make biology easier to engineer is to develop tools and platforms and technologies. And that's what a lot of the people who are in this industry are doing. And that is in turn going to then make drugs faster, better, and cheaper to produce. So that's why this is relevant. And then later on, we're going to look at some of the specific details of some of the companies that are involved in this industry and why they are doing what they're doing or what they're doing. So we're also going to cover some of the challenges that are involved in the biopharma industry. And we're going to look at some of the tools that have been developed for early stage research and development. In particular, one of the tools is gene editing, and we're going to look at CRISPR-Cas9 therapeutics and diagnostics. And we're going to look at a deep dive into non-native nucleosides and amino acids. And there's a couple of companies in particular, Ambrx and Synthorx, and I believe that they're both in San Diego. I know that Synthorx is. I think that came out of the scripts, if I remember rightly. And what you can do with that ability. So we also have some strategic recommendations at the end of the report that Mark is going to be giving us.
So yeah, well, as you were mentioning, this is part of the What's Your Biostrategy series that we've been doing, looking at different industries based on the book that you and Carl co-authored. And yeah, basically taking what's in that and applying it more specifically to different areas, in this case, biopharma. So John, you and I were talking the other day about the sort of mixed perceptions that are around biopharma today. There's a lot of concern at some levels about things like price gouging. There are people who are anti-vaxxers and afraid of traditional, or rather afraid of conventional medicine, and prefer home remedies and other things that sometimes are ineffective, sometimes work, sometimes are quackery, sometimes are dangerous. And it's a pretty complex field. And yeah, so was there anything from that conversation you wanted to bring back up?
Well, I agree. I think that there's two images that the biopharma industry conjures up. One is price gouging, the Martin Shkreli's of the world, and the other is the this particularly came up because in the opening paragraph of your report you mentioned the snake oil salespeople of the I think of the 17 or 1800s, and it made me laugh because there's still a lot of that going on. There's still people who buy, oxygenated water thinking that it has some health benefit. There are still people who buy certain kinds of homeopathic remedies or certain oils that they think have some properties. And it's really, these stories are really ingrained in people's belief systems around the world about what actually makes you healthy and what makes you better. and a lot of it doesn't align with the science. Then when you do get things that have to align absolutely with the science, it creates this very regulated industry that costs then billions of dollars to get drugs to market. And then once those drugs do get to market, those companies then have to recoup all of the costs of the development and all of the costs of the failures as well. And so if you look at it selfishly, then sure, too right, they want to charge as much as they can for that drug. And if it's a life-saving cancer drug and they know that people are dying, then they'll charge as much as they can get away with for that drug. So I think those were the two thoughts that I had and the two thinkings that I think frame both ends of this of the biopharma industry.
Yeah, and I think we're going to be addressing just about all those things as we talk. And I think it's also good to look historically how we got from snake oil to where we are today. And you know, this staff and the two entwined snakes that kind of look like DNA. You know, this has been a symbol of medicine for a long time, and the staff of Hermes. And the thing that I read into a lot of the symbolism here is the rooting in natural things. So snake oil and snake venom and other natural compounds have been used throughout millennia to find new cures for things. And it was really only recently, about 150 years ago, when pharmacology started to be a science, and thus snake oil. And the scientific method started to bring a lot more rigor to bear. And we're at a point right now where some of those natural remedies are actually the objects of synthesis of some of the synbio companies, the synthetic biology companies that we've been looking at. But also, as you're saying, the ability to know what works and to have data to prove what works is something that other companies are looking at. So better diagnostic tools, better lab equipment, and so on. But the real reason that this is so high on the political and social agenda, and why it's so politicized more than just about any other industry is because on the one hand it's about life, and that is a factor that people literally create religions around, and the other is the expense. It's a huge and growing expense, and that governments around the world and insurers in the United States are struggling to pay for, and individuals too. So if we can help bring synthetic biology to solve some of those problems, I think we can do a lot of good. So medicine is basically that practice that started with finding plants and animals and excretions and things like that gave some kind of an effect. There was a lot of quackery for a long time, but as that matured, we got into pharmaceuticals and specifically into biopharma. So to make a distinction between medicine generally includes natural medicines, pharmaceuticals generally are synthetic drugs, and biopharma is biological molecules, it's therapeutics. And so again, to make that distinction a little bit more fine-grained and clear, for most of the history of modern medicine, pharmaceuticals have been small molecule drugs. So this little bitty picture is a picture of the chemical structure of aspirin, acetylsalicylic acid, and the diagram there basically shows how these atoms are arranged. So 9 carbons, 8 hydrogens, and 4 oxygens. It's pretty simple, but it has a very powerful effect, and it's a very common medicine. Well, around the 1970s, we started getting the ability to take genes from one organism and put them in another. And in particular, insulin was the first biological therapeutic molecule. So insulin is a complex molecule. It looks like this. So same basic idea here with these lines connecting atoms in this overall structure. It's vastly more complex than aspirin. And so this wasn't something that could easily be synthesized in the lab. It wasn't something that if your body wasn't making it, you could easily get anywhere. So the way we got insulin before the 1970s and 80s was basically from cadavers, dead people, dead pigs, and dead cows. And it's something on the order of 50,000 cows' pancreases were needed to supply 1 kilogram of insulin. It's a very, very difficult extractive process. And since cows and pigs are slightly different from humans, it was a pretty bad it wasn't a perfect match. And so diabetes was often a death sentence because it couldn't really be treated with the insulin that we had at the time. So in the 70s, Genentech and 80s, Genentech basically commercialized the human insulin by taking the gene for that from people and putting it into a microbe that then they could grow and control really easily, and it would put out as much insulin basically as we needed. So even though this molecule is really complicated, we learned how to produce it at scale, at cost effectively, and everything else. So today, insulin is it's still not good to get it, but it's still much more manageable than it ever was in the past. So the difference.
And
oh yeah,
Mark,
go ahead.
Sorry to interrupt. We were talking about this yesterday when we were prepping for this webinar, and I said, well, you could argue, and Carl Handelsman at Genentech Roche Venture Fund does argue that Genentech was the first synthetic biology company. In 1968, when it was formed in South San Francisco. And there's this very famous photograph, which I was telling you about. And I said, well, do you know that photograph of Bob Swanson and Herb Boyer in front of the blackboard on the founding of Genentech? He said, well, I've seen that photograph with the blackboard. And I said, well, did you read what's on the blackboard? And you said no. So I said, well, if you look closely, so let's look closely at this blackboard. What you can see written there is the word synthetic DNA. And what you can see here is the plasmid, and here's the E. coli cell being opened up. And here is the insulin construct that's put into it. So I think, depending on your definition of synthetic biology, I think Carl is right. You could argue that Genentech was the first synthetic biology company. Okay,
well, we're going to have to rewrite their investor report to include Genentech now, but yeah. Well,
which is coming out tomorrow morning at 9 a.m. Pacific, by the way. Mark and I will be back for the investor webinar, which we encourage you to join us for too.
So in addition to their structural differences, there's obviously a huge difference in how these drugs are manufactured. And that incurs a lot of cost, a lot of complexity, new regulatory pathways, and a lot of other stuff that basically mean that these biomolecules, which are relatively newcomers to the field, are really a much higher growth area. They're coming from behind, so to speak. In other words, most of the molecules that we've made so far have been small molecule drugs. The vast majority of drugs on the market are small molecule drugs. But biomolecules are starting to take over. Not in count, the number of biomolecules approved as medicines is still pretty low, but they make a lot of money. They cost a lot of money to make, but you can charge a lot of money for them. So in terms of revenue, they're actually speedily creeping up on small molecule drugs in terms of revenue. So that should hopefully give you a good outline of what biopharma is versus small molecule drugs or traditional pharma. So if you look at the overall biopharma market, the first thing to know is that what were previously pretty separate industries, that is biotech and pharmaceuticals, are rapidly converging. So now I think it's fair to say that pharma and biotech, that it's red biotech, in other words, medical biotech, are pretty much the same industry today. You used to see companies making either one or the other, and now most companies, at least bigger companies, make both. So if you look at the global 500, this is the 500 largest companies in the world by ranking, by revenue, according to Fortune magazine. At the right here, you can see a list of the biggest companies in biopharma. So most of these companies probably have names that are literally household names that would be familiar to you, or if you're in medicine, that you'd be familiar with them as drug makers. So Johnson & Johnson, Bayer, those companies make medicines that you can buy over the counter and you probably have in your home. Roche and Novartis and Sanofi, Aventis and Merck and GlaxoSmithKline, these are all, again, traditional pharma companies. Some of them have had, like Glaxo has, been in consumer products and things like that. But for the most part, these are drug makers. So overall, spending on medicines globally is about $1 trillion a year, and it's growing at, a pretty, this would be considered pretty slow growth, 2 to 5% per year, compound annual growth rate. But that overall average and overall number hides some really interesting dynamics. First of all, there's a category of drug that we call blockbuster drugs, and those are basically drugs that generate $1 billion or more a year annually. So Vioxx and Lipitor and Zoloft, you've probably heard of some of these drugs. These are the drugs that most drug makers are trying to hit, get some really big revenue drugs, and they'd much rather have one big one than say 10 medium-sized ones. Another dynamic though is on the other end of the spectrum, and that's the revenue from a drug can drop like by 90%, essentially, close to zero when it goes off patent and generic makers will start to produce it, or when in some cases, a disease is eradicated or fully managed. And we've seen that happen even with some synbio drugs, like one of the initial drugs coming out of synthetic biology was artemisinin, produced by Amyris. And what's happened basically is because the market has really stabilized, there's not a need for the synthetic version as much as there was when they started making the drug. So these dynamics are really, really difficult to navigate. And I think the best thing that we can say is that synthetic biology is a great toolkit to add to both the biological and the small molecule drug side. And we'll see why as we go forward. Anything you want to say on that, John?
I think it's a good comparison to look at these billion-dollar drug companies or billion-dollar drugs with the craze for Silicon Valley to chase unicorns, really looking to go after these big problems with big solutions. And certainly when you can go after one of these and cure a disease or eradicate it, then you're gonna, they are big problems and they're after big solutions. I was blown away the other day because I read the statistic that tuberculosis is people, 1/3 of the world's population is infected with tuberculosis. Did you know that?
No.
Isn't that amazing? So talk about big problems. I mean, some of these health issues are huge. Now, unfortunately, it's the 1/3 of the people who don't have the money to pay for medicine. So it's not a big, exciting, sexy opportunity for a biopharma company, but there are certainly big problems out there that need solving with technology.
Yeah. Yeah, so before we go to the next slide, I also want to point out two companies on here that probably nobody's heard of, that's on this call anyway, China Resources and Sinopharm. So you can probably tell from the names, both of them, these are Chinese companies, and these are relatively new entrants. And so I'm going to talk about them in just a second, so I wanted to point them out though.
Yeah, and before you move on from that slide, Carl, Mark, apologies. It's also worth pointing out that nearly half of those biopharma companies are going to be at SynBio Beta. Roche, I mentioned, Carl Handelsman is going to be there. Bayer is going to be there. Novartis is going to be there. Merck is going to be there. GSK, we have John Baldoni doing a fireside chat on the future of machine learning in the biopharma industry. Gilead is going to be there, people from Cell Design Labs. So a lot of biopharma presence come into the conference and trying to figure out how they're going to use these new technologies and new platforms and new tools to make biology easier to engineer. Okay, so as I said, there's a lot of changing dynamics in the biopharma market. First, the merging of small molecule drugs and red biotech, but there are a lot of other players that don't make the top 10 or 20 list that I just showed you. So there are non-corporate actors, that would be governments and university research that really are a major source of innovation in the industry, and there are also startups and venture investors. So we're going to talk in a little bit about the cost of R&D, and one of the ways that the pharma companies have de-risked their own R&D is by basically letting startups do a lot of the risky work and then buying them up using a venture arm to invest in them and acquire them as they move from idea towards the market. So last year, the venture investment in pharma hit almost $17 billion. That's up from $12 billion the year before. So it's a very big rise. And it reflects, I think, a lot of optimism about some of the technologies that are coming to market, the diseases that they can help treat that we haven't had good treatments for, and so on.
The second aspect of that is emerging markets. So I mentioned again Sinopharm and China Resources. These are both extremely new entrants to the top ranking, and most of the names on that list have been around for 50 or 100 years in some cases. They're very old companies, and mostly based in North America and Western Europe. So the entrance of two new companies just in the last 8 years or so is actually pretty startling. And the fact that one of them is the biggest is certainly noteworthy. And this is due to a huge investment that China as a country has been making in advanced medicine. So the Beijing Genomics Institute, which does gene sequencing, is now the largest in the world. And it's really, the go-to place for a lot of the genomics work that's being done. So, so that's a notable change. And then third, as I mentioned, the blurring of pharma and biotech doesn't stop at the medical wall, so to speak. So Roche a few years ago bought Genentech for $47 billion. That was probably one of the bigger deals, getting biotech and pharma together. But there are all these other areas where, again, you have consumer products that in some cases are selling complete sham types of things. So cosmeceuticals, nutraceuticals are cosmetic and food-based therapeutics. But they, while there certainly are products out there that are in those spaces that make sense and seem to work also with natural remedies, and then would stand up to clinical trials. There are a lot that aren't, and they're just based on a lot of hype and who. So it's difficult because there's no real regulatory scheme that applies just as well to those products as it does to pharmaceuticals.
Just to jump back to the China market for a second, Mark, I actually, some inside knowledge from folks I know in China is that the BGI is actually now not the largest gene sequencing center in the world, that there's another one or two larger centers within China that are competing with the BGI and are now bigger.
That was really sneaky. That was smart. Okay.
And right. Then second is looking at this the Sinopharm or Sinopharm and China Resources. Do you have any idea whether they are licensing biopharma drugs from Western companies or European, American or European companies, or any outside of China, and then bringing them into China and servicing the local market? Or are they developing new drugs for the China market and servicing the China market and then looking to license them out? And I know you're not an expert necessarily on particular piece.
That's a good question, and it would actually not take expertise. It would take just a quick Google to find that out. But I would look at their portfolios and to answer that directly. But my suspicion, looking at what other China-based pharmaceutical manufacturers are doing, is there is a whole portfolio. Some of it's even generic drugs, some of it's licensed drugs, and some of it's new drugs. And I think the goal that, again, across the board that the Chinese industry has is to just keep moving up the ladder in terms of technology and more advanced applications. You see that in a lot of other areas we're not touching on today.
And as you and a lot of other people know, I am a China-phile, and I'm very excited about what's going on in China. And you're seeing this shift that I think you saw with Japan. In the 70s, Japan was producing cheap crap that nobody wanted. Now Japan is well known for producing well-designed, solid products, and they've moved from a copying manufacturing economy into an innovation economy. You're seeing the same in China. As one bullet point for the biopharma world, there's a company called Beigene, B-E-I-G-E-N-E, very cleverly titled, which is a company that's based in Beijing doing cancer immunotherapy. What's very interesting is that the technology comes out of Beijing, the company's in Beijing, and they have an office in Emeryville here in California to do the clinical trials, because the world still sees the US as the clinical trial standard, and then they'll get the clinical trial approval, and then they'll take the drug back to China. So I thought that was an interesting model as well.
Yeah, absolutely. And that's a good segue to the next slide, where we're actually talking about that process. So this is a simple version of the biopharma industry value chain. Obviously, you can make much more complex versions of these, and a lot of people do. But the basic process is this: you go from discovery, which is mostly, chemistry, you could say, basically trying to find candidate molecules, and you might screen millions of molecules for properties that you think would be worth actually testing in animals or even on cells. So once you get a drug out of that initial screening process, the next step is to, again, put it on cells or in animals, and those experiments start to be more expensive. But you're looking for basically, does it hit the protein that we're targeting? Does it seem to affect the condition that we want to treat with this drug? And if it does, and it looks like so it's effective, it's got the right things like solubility and manufacturability and so on, then it'll move to the phase 1 trials. Now, there's phase 1 trials are basically healthy human volunteers who who get paid to take a very small dose of the drug to make sure it doesn't have any unforeseen side effects that wouldn't have shown up in animals. And there's actually a pretty low translation of success rate from animals to humans, which is why before you go into a big study with people that are actually suffering from the disease, you want to de-risk it as much as possible. Again, there's pretty low translation of success. So all that screening that we've gotten up to now for millions of drugs, now we're down to still a few thousand drugs probably per year that actually go into phase 1 trials. Even there's a pretty low success rate. So overall, from phase 1 to approval, only about 12 to 18% of drugs make it through that whole process of human trials. But after phase 1, something like 75% of the drugs pass. In other words, those healthy volunteers can tolerate it. Phase 2 is where things really crater. Most drugs, only about 33% of drugs or less that go into phase 2 come out of it. In other words, they are shown to be safe and effective in humans. They're shown to have some beneficial treatment on animals, but humans don't react in the same way. So that means that the vast majority of drugs that we've been trying up to this point, only a small few get through phase 2. And this is really the key problem in drug development. And our question for today is, can synthetic biology fix this? Some of the numbers around what it costs per drug is something like $1 to $2 billion and maybe 10 years to get a drug through this entire process. So if synthetic biology can help with better tools for screening, or better animal models, or even humanized organs, things like that, that'll help the early stage trials be more effective at screening out compounds that don't work, then the later stage trials, where we're bringing in people in phase 2 to see if it works, and phase 3 to find the right dosing, will be more effective. Anything you want to add there, John?
Well, I think this is now getting to the meat of the webinar, which is that we suck at engineering biology. Biology is complicated, and that these are dismal failure rates that go from initial product to approved drug. And so I'm excited for the next part, where you're going to show us what all these new innovative synthetic biology companies are doing about it.
Yeah, so basically as part of the study, what we did is we looked at, this value chain problem is really critical. It affects, as we said at the outset, it's political, it is social, and it affects a lot of how we think about healthcare and life. And there was a study that we looked at while we were doing the research for this, that was done by A.T. Kearney, and they basically make the argument in quite a lot of words and some diagrams here, that the early stage, pre-phase 2 work, clinical proof of concept, is basically where you do a huge narrowing down, and it's really distinct from the later stage phase 2 to market part of the process, where you're looking for, again, you're looking for things like, what's the right dose, what's too much, what's too little, is it a twice a day or once a day or 3 times a day type of regimen, what should the price be, and some other things like that. So it's much more of a business and human related process, whereas upstream is a lot more about science and technology and things like that, and of course biology itself. So they propose a kind of a split, and they asked, what would it be like if the pharma value chain split right at, inside of phase 2. So you have, there are different phases within phase 2, A, B, C, and so on. And you basically say upstream of that, you'd have disease-focused research units or science-driven drug discovery units. This is where the genomics and medicinal chemistry and pharmacology would come into play for initial like toxicology and so on. Once things got past that, then they'd go into a very different type of business where once things actually clear phase 2, they do a pretty good job of seeding. And so that would be a lot more about clinical study management and regulation and so on. So if that were to happen, the places that most synthetic biology startups would find themselves is in this upstream phase. And so they could arguably play a much more important role in pharmaceutical companies' portfolios than they do today. And this is critical because right now, the way pharma usually works is they will step in phase 2 and later. And that's when they'll buy a company that's actually shown all of that work to work out. But by investing actually in that upstream activity, they might actually improve not only their upstream performance by the drugs that they do keep in-house, but also by looking at downstream, having better screens and better tools that will help them make better choices about what they invest in later stage. So if we can use those types of approaches to get at this, $1 to $2 billion per drug problem that we've got, then that would be a huge impact. And synthetic biology, well, sorry, John, did you want to say anything about that?
No, we have an interesting question kind of related to this. Do you want me to give it to you now, or do you want to finish what you were saying?
No, go ahead.
So it's Jack Kenny. And Jack Kenny says, given a high proportion of total development costs are tied up in the discovery phase, can you elaborate on examples of how synthetic biology, as well as computational biology, may be applied to accelerate and better target drug discovery? Do you perceive these techniques to have more applicability to small molecule development or biologic development?
Yeah, well, first of all, yes, so both of the things we're going to go into in a little bit of detail in just a second here. Looking at some of the specific companies in synthetic biology that are looking at exactly those stages. So yes, computationally, it's great if you can model things in software and basically see what's likely to have the desired effect and impact that you've got. And I just heard something today. I'm in Denmark right now, and I heard something today that I haven't been able to directly verify. But supposedly, I was told that Novo, the company, Novo Nordisk, big diabetes and other pharmaceutical company, and one of the first biopharmaceutical companies, has laid off 400 people in hematology because they feel like that market is just not growing, and what they're hiring is quantum computer experts. So basically, people that can help them do quantum modeling of molecular interactions and the types of things that really do come into play in a big way in biomolecules because they're so much more complex than small molecules. So like I said, I don't know that that's true. It's just a rumor, but if it's true, it says a lot about where this upstream research is going. Actually, they'll probably make it one of the biggest investments in an application of quantum computing to date. Almost all the other investments in quantum computing have been by companies that are in computing, so Intel and Microsoft and so on. And in this case, it would be an application. So yeah, I think we're really only beginning to scratch the surface around how bioinformatics and computational biology can help not only screen drugs upstream, but also even come up with new permutations and configurations and things like that. So new variations on molecules and see how they might react.
And another question is, can and should we split the value chain at phase 2? It sounds like we could.
Yeah, so the argument, I mean, it's not a moral argument, I guess, it's a financial argument. And I think it rings true to me that there's a really detailed analysis in this Kearney document, and you can see the link at the bottom of the slides that I think are going to be distributed. It's also in the report. That it's a good argument, and you could see a much more effective pharmaceutical industry come out of it if that actually became the case. So I think it sounds like a good trial, something to give it a shot.
Great.
Okay. So back to synthetic biology, you know, why this is so important. There's probably zero other fields right now that have as much potential, much promise for biopharmaceuticals as synthetic biology. There's not some other like way of thinking about disease or about biology that has as much to recommend it. And so the very problem that keeps biopharma running is this technology advancement. In other words, we keep getting new ideas, new ways to approach the treatment of disease and diagnosis of disease, but we don't always have the sort of accompanying tools like the lab tools and the animal models or the cell models to test those things. So one of the things that you see when you look at the synthetic biology field is there is this sort of repeat success history that so many people have. Some of the giants in the field like George Church and Jay Kiesling, these are people that have been behind more than one company that have come up with some really amazing solutions. Or Tim Liu is the MIT Synthetic Biology Group, part of their core team. And just the number of people that one researcher and their lab can be part of co-founding is really impressive. And so, for that reason, I think it's not just that these people are so darn smart, although they certainly are, it's that this is just a really rich platform for innovation and for experimentation. And so I think that there's a strong reason to focus on it for that as we look at this upstream, downstream value chain problem. So the rest of the presentation is basically going into some depth about what companies in synthetic biology are taking, what types of approaches, what they're working on. If we wanted to, I think we need to be done in about 10 minutes. And so we unfortunately, I don't think would make it through all of them. And we do want to leave a few minutes at the end for questions and answers. So I think what I'll do is I'll present the first slide, ask you, and then see where we go from there.
Sounds good.
Okay. So we talked about early stage R&D, and there are quite a number of companies, just 3 listed here, but a lot more that are looking at tools for, computational biology, for lab automation, and a lot of other things. The 3 that we wanted to point out here are A-alpha Bio, which is creating engineered yeast cells to measure a lot of different, thousands of protein interactions simultaneously in a single sample. And, if you've ever been in a lab and had to do something like that, basically the division of a sample into small enough individual units so that you can test a lot of different things can really put limitations on your experiment. If you have a really small sample, it's difficult to get a sample. Dividing it up consistently and then treating it consistently and getting a readout can be a lot of work. So being able to look at thousands of interactions simultaneously in one sample is a pretty big leap forward just in time and cost saving.
And I believe that A-alpha Bio is going to be speaking at SynBio Beta, and also it's a spin out from David Baker's lab at University of Washington, I believe.
Okay, great. Atomwise, we talked earlier about computational biology. So this is a company that's using AI to screen drug candidates. They claim that they can do 10 to 20 million compounds a day, and they've got some of the big players in pharma that we mentioned earlier as their partners. So that is probably also vouching for their capabilities. And then there's CRISPR. So CRISPR we'll talk about in a little bit, but gene editing technology or technique that's been described as a new era in biopharmaceuticals, and basically using that as a method for doing drug discovery. What this company's doing. So really, again, far upstream, these are things that don't even go towards into animal testing, just looking at really early stage R&D. But then you can also look at some companies that are actually synthesizing active molecules. These are generally small molecules for testing and production. So some of this would be if you want to test a candidate drug without, and it's difficult to synthesize, sometimes you can put that into a model organism and then produce it for testing. But in some cases, these are also producing actual therapeutic molecules that are difficult to harvest or to produce. So opioids, for example, Afghanistan, poppy seeds, dangerous, drug trafficking. It's very difficult, a lot of times to get good good quality and large scale amounts of drugs like that. Cannabinoid is another molecule that, due to restrictions in the United States and other places on production, you can't grow marijuana and extract just that plant. That's a difficult regulatory pathway. So by just producing the active molecules, Renew Biopharma is trying to do that. So there are a few other examples here, Dimitrex and Oxford, that are trying to increase the use of synthetic biology, increase the production of biomolecules and therapeutics through synthetic biology.
Excellent. And it's worth noting that two of those, I think Dimitrex and Renew, are both making THC derivatives, cannabinoids. I think Dimitrex is also doing that. Great. What's next?
So we've got CRISPR-Cas9. I think what might be good, since we're just about 5 minutes left, I'll briefly just go through the rest of these, and then we'll get to kind of an endpoint, and then we can have a conversation. So we were also looking at CRISPR-Cas9 for therapeutics and diagnostics. There are a number of companies doing really interesting things, like Mammoth is using CRISPR to develop proteins that they can basically use for diagnostics. There are alternatives to CRISPR, like Thermo Fisher has Talons and Intrexon has Sleeping Beauty. So there's a lot of different ways to do gene and genome editing. One of the areas I've always thought is really fascinating is the use of non-native nucleosides and amino acids to create new kinds of genes and proteins that you wouldn't find in a natural organism. So they're basically adding new letters to the alphabets of genes, which would be DNA, or proteins, which would be amino acids, and those sometimes have better pharmacokinetics or some other performance benefit. But sometimes it's also just a way to make sure that those biomolecules can't be produced outside of the lab environment. The microbiome remains, a huge topic of innovation. So there's, far more companies than we can put on the slide, but companies are basically looking at modifying individual members of the bacteria, bacterial members of the microbiome to achieve certain benefits, or to look at the overall makeup consortium and how that can be tweaked to treat different diseases of the skin and of the gut and so on. And then finally, just to wrap this up, the 3 recommendations that we have, looking at what's going on in the field of synthetic biology, the needs that the biopharma industry has, and how those two can come together. One, as we were just mentioning, there's a lot of in-house research in terms of skills that can be expanded by using the people and tools that are emerging from synthetic biology. One of the things that's really interesting about synthetic biology is because it is relatively new and different compared to a lot of other subdisciplines within bioscience, is it attracts interesting people, and it attracts people that often work across the boundaries of their skill, of their domain. So you might have somebody who's working on biopharma, but they've also had experience in industrial biotech and agriculture and so on. The partnerships with startups in the synthetic biology space is one, again, we've already seen has a lot of benefits. Here too, the fact that synthetic biology really extends outside of biopharma is a pretty big benefit, because that means you can spread the investment and spread the risk with non-competing companies that might be making, say, bioplastics or treating crops. And then finally, for biopharma, the industry sort of gets more or less aggressive or ambitious, and then sort of ratchets it back as the regulatory and other cycles wax and wane. But there are a lot of areas that synthetic biology is developing, like organ replacement, for example, cell therapies, organ replacement, we didn't really touch on very much today. And even things like life extension, being sort of the ultimate collective solution to problems of disease and health. And those are areas where other companies that are really focused on, well, even like Google and companies like that are starting to try and take leadership positions. So in the longer term, this would be something for pharma to look out for. As more and more contract manufacturing becomes available, companies will be able to tie into that, even if they don't have a long history in the biopharmaceutical supply chain. So to keep
Excellent. Well, we have come to the end of the webinar. Thank you for joining us, Mark. We have one question from the audience, which we'll come to in a second. I want to remind everybody that we're doing the investment report tomorrow, and we have another webinar with Mark and I, and you'll be able to read that report at, I believe it's 9 a.m. tomorrow. We're going to be live again. So the last question, oh, and I wanted to say that we have some very exciting sessions on biopharma. In particular, we talked about some of the engineering the microbiome, and we have a session that's sponsored by Ginkgo Bioworks on living medicines and engineering the microbiome, and a lot of interesting speakers in there. In particular, Synlogic and Ginkgo announced a partnership. Synlogic's not speaking at the event, but they did announce a partnership all about living medicines and how Ginkgo is creating engineered microbes that Synlogic is using inside the gut. So very exciting topic. And we also have the fireside chat with John Baldoni and Mark Fisher-Colpreet and Aishani Shatra, who are going to be speaking about machine learning and biopharma. So a lot of exciting sessions, and another one from Oxford Genetics, who's sponsoring a breakout session on molecular tools for biopharma. And most importantly, one from Sandor Shoiket, who's going to be doing a session on what's your biostrategy for the biopharma industry. And that's actually where the question comes from now. Sandor is watching live. And Sandor says most of the cost is actually in the clinical development phases 2 and 3. He says reducing the clinical failure rate would be the biggest impact. Are there companies already focusing on this, Mark?
Yeah, so going back to what I was saying about the failure rates from different phases, from screening to in vitro studies to in vivo studies and so on. There's a pretty big drop off in each of those. But as you say, you're still all the expense comes in when you start to have human trials. Those can take years and thousands of patients and a lot of people. So there's two ways to basically look at improving that math. One is to not let so many drugs into clinical trials that don't have a chance of making it. So there are companies in synthetic biology that are looking to do things like engineer pig organs so that they are compatible with human immune systems and that they could basically be also used for testing. So a lot of basically coming up with better models of human organs and human systems is one thing. The other one is upstream. We have all these millions of candidate molecules that we screen, but there's probably, there's still, an effectively infinite number of other ones. And so by essentially coming up with more candidates earlier, which synthetic biology can help with, there will be more, more positive shots on goal to take, basically.
Great. Well, excellent question. So thank you all for joining us. And if you're interested in the investor report tomorrow, which is going to be some very exciting news showing the biggest growth in investment for the since we started tracking the investment data, then please join Mark and I tomorrow morning at 9 a.m. Mark, thanks again for joining us live from Denmark, and we'll see you tomorrow.
Okay. Thanks, everyone. Take care.
Cheers. Thanks, everybody.